Welcome all you for this lecture called Special Types of Genes. So in this genes we are about to see about structure, function and arrangements of the genes which will differ in these aspects, and also some special types of genes called split genes, overlapping genes, pseudogenes, oncogenes and other gene families and jumping genes. So these are all playing very vital roles in the plant evaluation and adaptations and crop improvement. So about the split gene, it is also called as an interrupted gene, is a gene whose coding sequence is not continuous in the DNA. So the coding parts, that is called exons, are separated by the non-coding parts, which is called introns. When the gene is transcribed, the entire sequence, that is introns plus exons, is initially copied into a precursor, which is RNA, pre-mRNA or hnRNA. So then during RNA processing, the introns are removed, that is spliced out, and exons are joined together to produce a mature mRNA that gets translated into a protein. So these introns are spliced out, which will function as alternative splicing, which is more protein diversity. So there are types of split genes based on the normal sequence and as well as the interrupted sequence. So overlapping genes means these are all the genes whose nucleotide sequence share the some region, that is part of DNA or that RNA transcripts is used by more than one gene. So these overlaps can be in the coding regions or in the transcripts. In prokaryotes or the viruses, the emphasis is usually on the coding sequence overlap. In eukaryotes, it is often transcript or non-coding region overlaps matter. So for example, bacteriophage, SV40 virus and PSPD virus genes are the examples of overlapping genes. So pseudogenes is a segment of DNA that resembles a functional gene in sequence or maybe in the structure, but it has lost its ability to produce as a functional protein, which means it is not a functional genes, it may be resemble as a functional gene. So this loss of function is maybe due to the disabling mutation such as premature codons, frameships, or loss of regulatory frameworks or the stop codons themselves. They are sometimes called as genomic fossils because they represent once the functional genes that are now inactive. So examples of these non-functional or pseudogenes or resistance gene pseudogenes are cytochrome P40, P450 pseudogenes in Arabidopsis. Oncogenes is a gene that has a potential to cause a normal cell to become a cancerous cell. That means overgrowth. The normal form of such gene is called as a proto-oncogene, which is involved in regulating the cell growth or division or apoptosis, which is a programmed cell death. When a proto-oncogene is mutated or overexpressed or abnormally activated, it becomes the oncogene. Example of oncogene is IAAM and IAAH genes and IPT genes or crown gall tumors in the daikons. So the gene families is a group of genes in a genome that are similar in sequence and functions and are derived from the duplication from a common ancestral gene. So MD box are the gene families, and gene cluster refers to group of two or more genes located close together in a chromosome that encodes similar protein or functional RNAs. So jumping genes, this is very important because it is also called as a transposons or transposable elements. These are all the genes that are segments of DNA that can move from one location to another location within the genome. This movement can occur within the same chromosome or in a different chromosome, which causes a genetic changes. So it is discovered by Barbara McClintock Mays in 1940s. She won the Nobel Prize for this in 1983. So its scientific name of these jumping genes is transposable elements or transposons. So function of these jumping genes, it can alter the gene expression or create the mutation and can contribute to the genetic diversity. And it is found in prokaryotes as well as eukaryotes. So there are types of genes which is a class 1 and class 2 transposons, which is significant in major causing mutations and gene rearrangements, useful in gene tagging and is genetic engineering, and also it contributes evaluation by promoting genetic variation. Example was in transposon in maize seed colors, ACDS elements in maize causes changes in kernel color by inserting into or excising from the pigment producing genes. So, in conclusion, these are all the different types of genes found in the genetics family: split genes, overlapping, pseudo, onco, and transposons, which majorly illustrate this complexity and adaptability of the genomes, which majorly contribute to the genetic diversity, which is a major diversity forming for the evolutionary process and also in disease mechanism and resistant mechanisms, etc. So understanding their role is very important to enhance the insight into the genetic diversity and also functional genomics and to the applications in the biotechnology and as well as the medicine. Thank you.